mention the plasma protein and its three important functions. So the plasma proteins are albumin, globulin and fibrinogen. Albumin, the normal levels are from 3.5 to 5 grams per deciliter. Globulin 2.5 to 3.5 grams per deciliter, and fibrinogen it is 200 to 400 milligrams per deciliter. Globulins are of alpha 1 globulin, alpha 2 globulin, beta and gamma globulins. Now we will see what are the functions of plasma protein. So albumin, it maintains the major function of albumin is to provide colloidal osmotic pressure in the plasma. So it will prevent the loss of plasma from capillaries and control the distribution of water in the body. Then it has got transport function. Albumin will transport bilirubin, free fatty acid, calcium ions and metals like copper and zinc, then steroid hormones and a variety of drugs. So mainly it will transport substances which are insoluble in water. That is, albumin is the carrier of various hydrophobic substances in blood. Then the other function of albumin is its buffering action. All proteins, they have buffering capacity. Because of its high concentration, albumin has the maximum buffering capacity. And albumin, it has about 16 histidine residue which will contribute to this buffering action. Then albumin also has nutritional function. That is, it is considered as the transport form of essential amino acid from liver to the extrahepatic cells. Then gamma globulins, they play an important role in the defense mechanism of the body. So these molecules, they will act as antibodies and they are known as the immunoglobulins. So the antibody, they will react with antigen of disease causing microorganism. So in addition to gamma globulin, beta 2 microglobulin, they are also involved in the defense mechanism. Then globulins, they are also involved in number of enzymatic functions in the plasma. Example as blood coagulation factors. Then fibrinogen, it is essential for the coagulation of blood. That is, during coagulation of blood, fibrinogen will polymerize into long fibrin thread, thereby forming a blood clot. So that will help to repair leaks in the circulation. Question is electrophoresis. So the term electrophoresis, it refers to the movement of charged particle through an electrolyte when subjected to an electric field. Here the positively charged particle, they will move to the cathode and negatively charged particle will move to the anode. And electrophoresis, it is a separation technique and it is used for separation of proteins in biological fluids. The rate of migration, that is the rate of separation of particle, it depends on the following factors. That is the net charge on the particle, pH of the medium, temperature, mass and shape of the particle, strength of the electric field and properties of the supporting medium. In agar-gel electrophoresis, normal serum is separated into 5 bands. That is albumin, alpha 1 globulin, alpha 2 globulin, beta and gamma globulins. Albumin has the maximum mobility and gamma globulins has the minimum mobility. Now the abnormal patterns. So in multiple myeloma, that is in paraproteinemias, a sharp spike is noted and it is termed as M band. So this is due to the monoclonal origin of immunoglobulins in multiple myeloma. That is, you can see an M band between the beta and gamma regions. In case of chronic infection, the gamma globulins are increased. So there is a broad increase in the gamma region and also a general increase in the alpha 1 and alpha 2 bands. Third one is nephrotic syndrome. So in nephrotic syndrome, hypoalbuminemia, that is the albumin band is decreased and you can see a prominent alpha 2 band. That is, all proteins except very big molecules are lost through urine. So alpha 2 fraction will be very prominent. Third one is cirrhosis of liver. In cirrhosis of liver, albumin synthesis will be decreased. So the albumin band will be decreased. Since the albumin synthesis is decreased, there will be a compensatory excess synthesis of globulins by the reticuloendothelial system. So albumin band will be thin with a wide beta fraction. So here albumin band will be thin and the beta fraction will be increased.